Today we're going to look at a different type of photosynthesis where photosystem 1 acts independently from photosystem 2. And this is called the cyclic pathway, and we're going to see why that is. So looking at this diagram, we see that this looks a little different from the linear pathway that we recently learned about. In this case, if we look here and we have photosystem 2 isn't involved in this pathway. All of the action is happening here with photosystem 1. So instead of electrons moving from photosystem 1, we would think it would come to ferredoxin here, and then electrons would go and reduce NADP plus to NADPH. That isn't happening in this case. Instead, we have ferredoxin taking the electrons from photosystem 1 and bringing them to PQ, so plastoquinone. So it's going to be continuously reduced by collecting the electrons. And the important thing is it is also taking hydrogen ions and dumping them into the lumen after it takes the electrons to the cytochrome complex here. Okay, so we're going to see the electrons continuously being moved in this cycle. This is important because it allows the system to build up a hydrogen ion concentration on the lumen side. It is not going to be producing NADPH. So plastoquinone, PQ, keeps moving protons across the thylakoid membrane, as mentioned earlier. Water is not being split by photosystem 2. So this is a good system to use when water is not so much available. Also, as mentioned earlier, NADPH is not being produced by this system. However, ATP is still being made via chemiosmosis through the ATP synthase complex. So that is the important thing to remember. We are not splitting water. We are not producing NADPH, but we're still getting the hydrogen ion concentration difference across the thylakoid membrane and still able to make ATP. So why do we do this? Well, we don't let plants do this. So this form of electron transport is favored when oxygen levels are low or not available. It's also used under normal conditions. So the Calvin cycle, which we're going to learn about next, requires a higher amount of ATP compared to NADPH. So this allows it to create a larger amount of just ATP. And other reactions that occur in the chloroplast require ATP. So this pathway allows that higher amount of ATP to be produced. So let's look at an animation that is showing what would happen. So this is essentially just taken from our linear pathway, and then I adjust it for the cyclic. So we have plastocyanin bringing an electron over to the photosystem 1, and we still need a photon of light, and we have the accessory pigments being energized and sending energy, not electrons, into P700, which is chlorophyll A, which best absorbs light at 700 nanometers. So this becomes P700 star, so energized. The electron is energized, and the electron gets passed to the accessory, the electron acceptor, the primary electron acceptor, which then becomes negatively charged, and at the same time, P700 becomes positively charged, and there can take an electron from PC. Now, instead of ferredoxin being over here and then sending electrons to NADPH plus, the primary electron acceptor sends them to ferredoxin over here, which will cycle them back through. So P700 becomes neutral again because it was positively charged and took an electron, And ferredoxin is going to take that electron, and the primary electron acceptor becomes neutral again. This allows for the electron to again end up being passed to P700 after it gets activated, and sends an electron to the primary electron acceptor, reducing it, and P700 becomes positively charged. And then take another electron. And so we're seeing them cycle this way. Ferredoxin sends its electron to PQ, the primary electron acceptor sends its electron to ferredoxin, and so on and so forth. So remember, PQ is also dumping hydrogen ions into the lumen, again creating a proton gradient, a concentration difference, as it sends an electron to the cytochrome complex. So for every electron it takes from ferredoxin, it dumps a hydrogen ion into the lumen. So just watching, we see the electron is able to be sent around in this cycle, and we have a hydrogen ion being sent into the lumen. Now, chemosmosis is still going to occur as it does in linear transport photosynthesis or the electron transport chain, as we have a low hydrogen ion concentration in the stroma and a high hydrogen ion concentration in the thylakoid lumen. And so the hydrogen ions are able to go through the ATP synthase, producing ATP just like in the other system. Okay, so this is cyclic photosynthesis, where we get only ATP being produced. We don't have water being split, and we do not have NADPH being created.